Hello friends, welcome back to my YouTube channel, that is BiologyCenter. Today I am going to teach you carbohydrates. Okay. In the previous lecture I have stated that in a cellular pool different kind of biomolecules are found. Okay, some are called micromolecules and some are called macromolecules. Okay, so today I am going to teach you macromolecules, that is carbohydrates. Okay, now if our body use carbohydrate as an energy source, okay, then it will yield the energy or release the energy, that is 4 calorie per gram. Okay, please do remember it gives the energy, that is 4 calorie per gram. Okay, but in our cellular pool, okay, in our cellular pool carbohydrate contribute about to, please do remember, it contribute about to 3% of weight of protoplasm. Please do remember it contributes about to 3% of weight of protoplasm. Okay, now let's see the definition of carbohydrate. Okay, as the name indicate or name itself, it is the hydrates of carbon. Please do remember it is the hydrates of carbon. Okay, so we have to simplify this. That will be, that is CnH2O whole n. Okay, now but when we simplify again, that will become CnH2nOn. Okay, now this is the general formula of carbohydrates. Okay, now let's see, this is the general formula of carbohydrates. Okay, now please do remember n should be greater than equal to 3. Okay, please do remember n should be greater than equal to 3. Okay, now let's see some monosaccharide that will start from 3 carbon. Okay, if the carbon is 3, please do remember, let's see number of carbon. Okay, here written there is number of carbon 3, 4, 5, 6 and 7. Then what kind of monosaccharide it will form? Please do remember monosaccharide. Okay, now let's see. If the carbon contains 3 carbon, okay, then the monosaccharide is called, that is triose. Okay, if it contains 4 carbon, then it becomes tetrose. If it contains 5 carbon, okay, then it will be, that is pentose. In the same fashion, it is called hexose, and the next one will be, that is heptose. Okay, all these are monosaccharides. Okay, now let's see, let's come to the general formula. Okay, this general formula are sometimes, okay, after some times it is discarded. Please to remember discarded. Why? Because for example, let's see, that is acetic acid. This is acetic acid. Okay, when you simplify this, then it becomes C2H4O2. Okay, this is non-carbohydrate. Please you remember this is a non-carbohydrate. Okay, but still it obey this general formula. But still it obey this general formula. Okay, but when you're talking about a carbohydrate, that is deoxyribose sugar, please do remember deoxyribose sugar, that is, okay, this is the deoxyribose sugar. Okay, that is C5H10O4. Okay, this is a carbohydrate. Please do remember this is a carbohydrate, but still it is being a carbohydrate, but do not obey this general formula. Okay, it do not obey this general formula. Okay, now due to which it creates a lot of confusion. Okay, that's why the standard definition come into existence. Okay, what are the standard definitions? Let's see here. a standard definition. Okay, now it is called the polyhydroxy. Please do remember that is polyhydroxy of aldehyde or ketone. Please do remember, if the polyhydroxy group contains aldehyde in it, then it is called aldose. Please do remember, and if contain polyhydroxy group with ketone group, okay, then it is called ketose. Okay, now let's see. Okay, now the simplest carbohydrate is that is 3 carbon compound, that is triose, that is called glyceraldehyde. Please do remember, the smallest or simplest monosaccharide. Okay, carbohydrate is that is glyceraldehyde. Please do remember. This is the structure of glyceraldehyde. Please do remember. Okay, now that is 3 carbon compound. Okay, it contains aldehyde and it is also contain polyhydroxy group. Okay, so this fulfill the criteria of a standard definition. Okay, that is polyhydroxy, aldehyde or ketone. Okay, now let's see. This glyceraldehyde are the reference molecule in which the all the carbohydrates are compared. Okay, now at the last carbon, if the hydroxyl group are present in right hand side, okay, that's why this is called D form of glyceraldehyde. Okay, that's why this is called D form of glyceraldehyde. Okay, now let's proceed further. Now we will talking about a hexose, that is 6 carbon compound monosaccharide. Okay, now let's see monosaccharide. That is 6 carbon containing. Okay, first of all we have to know, we have to make 6 carbon. And 6. Okay, now this is the structure of glucose. Okay, now how this is the structure of glucose. Okay, now how do you know? Please do remember. Let's see. As I was stated earlier, in a glyceraldehyde, okay, that is the last carbon. From the last carbon, if the hydroxyl group is present right hand side, that is called D form of glucose. Okay, so please do remember, this is also a D form of glucose. Okay, now you have heard the name of DDLG. Okay, you have seen the movie DDLG, Dilwale Dulhania Le Jayenge. Okay, in the same fashion, you have to learn simply DDLD, that is Dilwale Dulhania Le Denge. Okay, and what kind of this? That is glucon D, D form of glucose. Okay, that is DDLD. Okay, that is D hydroxyl group present right hand side, DLD. Okay, DDLD. Please do remember. Mark this point. Okay, now let's see the L form of glucose. Okay, mirror. When talking about it, that it is a mirror image. Okay, let's see. Now this is the L form of glucose. Okay, let's see. Hydroxyl group are present in left hand side. Okay, now when you're talking about a monosaccharide, then it contains aldehyde group in it. Okay, in case of glucose, but when you're talking about the fructose, then it contains ketone group in it. Okay, let's see here. In the second position, in case of fructose, in the second position it contains ketone group. Okay, please do remember. But when talking about glucose, then aldehyde content in first carbon. In the first carbon aldehyde present. Okay, now this is the structure of fructose. Okay, now what do you mean by epimer? Let's see here. Epimer. Okay, now which compound the configuration is changed in a single carbon. Okay, then it is called epimer. Let's see. For example, that is mannose. Let's see. This is the structure of glucose. Okay, now let's see. Okay, now mannose are the epimer of glucose at the position of 2 carbon. Okay, please do remember. Let's see here. The epimer of 2 carbon. Okay, the configuration is changed in 2 carbon only. Okay, that's why this is called mannose is the epimer of glucose. Okay, now but I'm talking about a galactose. Okay, then it is epimer of 4 carbon. Let's see. All the structure are same, please do remember. Let's see here. All the structure are same, but changing the one position of a carbon, that is called epimers. Okay, this is called galactose. It is also 6 carbon compound. Glucose is also a 6 carbon compound, and mannose is also 6 carbon compound. So how do you learn? Simply you have to learn man nose. The man has one nose, has 2 nostrils. Okay, so the epimer on 2 carbon. But when talking about galactose, it is epimer of 4 carbon. Okay, now let's see. Now this is all the linear chain of monosaccharide. Okay, now this linear chain form a ring structure. Okay, let's see the ring structure of monosaccharide. Okay, now we are talking about the glucose. Okay, then it form a ring structure called pyranose. Paranose that contains 6-member ring. Please do remember. Contain a 6-member ring. It means that it means that contain 5 carbon plus 1 oxygen. Okay, this is how. But when talking about a fructose, please do remember fructose. Okay, then it form a 5. Then, for example, let's see fructose. Okay, then it form a ring structure that is called furanose. Okay, please. That is called furanose. That is, as the name indicate, 5-member ring. It contain 5-member ring. Okay. That is contain 4 carbon plus 1 oxygen. Okay, let's see how it is formed. Okay, now first of all we have to make a glucose. This is the structure of glucose. Okay, now in the last carbon, above the last carbon of hydroxyl group, it react with aldehyde. Okay, react with aldehyde. That reaction is called hemiacetal. It is called hemiacetal. Okay. Now, and form a ring structure. Okay, so glucose exits in 2 forms. Okay, it exit in 2 forms. Okay, if the hydroxyl group are opposite to CH2O group, CH2OH group, then it is called alpha form of glucose. Please do remember, it is called alpha form of glucose. But when talking about the beta form, that is hydroxyl group is same side of CH2OH group. Okay, that is called beta form of glucose. Okay, now. But when talking about a fructose, let's see here. Just like a glucose, the last carbon hydroxyl group react. Okay, so it form, so it becomes OH. Okay, now this ring structure is called furanose. Okay, now and it forms just like this. Okay, now let's see. Now the monosaccharide is over. Now come to disaccharide. On hydrolysis, when we hydrolyze, on hydrolysis, it gives 2 monosaccharide. Okay, for example, let's see here how the disaccharides are made. Okay, we're talking about the 2 glucose. Okay, now the first glucose bond with the fourth carbon of second glucose. Okay, in which water molecules are removed. Okay, now let's see. Water molecules are removed. Okay, this is called dehydration or condensation reaction occur. Okay, condensation reaction occur. Okay, now this linkage is called an glycosidic link, glycosidic linkage or bond. Okay, and form a glycosidic bond. Like that. Okay, now this is the glycosidic bond. Okay, now let's see the bond between, that is, both are the alpha form of glucose. Okay, so you have to write down alpha, that is 1,4 glycosidic bond. Okay, now the disaccharides are reducing in nature. Why? Because let's see, first glucose or first carbon is involved in bond formation, but when you're talking about second glucose, that is aldehyde, is free. Okay, that's why disaccharide is reducing in nature, that is reducing sugar. Okay, for example, let's see here, we're talking about maltose. Then when we hydrolyze this maltose, then it will break into glucose, 2 units of glucose. Okay, now in the same fashion talking about a lactose, but on hydrolysis it gives glucose plus galactose. Okay, but when you're talking about a sucrose, okay, then it will gives glucose plus fructose. Okay, please do remember and mark this point. Sucrose is non-reducing sugar. Non-reducing sugar. Mark this point, this is very, very important. Okay, now let's come to oligosaccharide. Oligosaccharides. Okay, on hydrolysis it will gives 2 to 10 monosaccharide. 2 to 10 monosaccharide. Okay, that is called oligosaccharide. But when you're talking about a polysaccharide, please do remember, but when you're talking polysaccharide, then it gives, on hydrolysis it gives more than 100 to 1000 of monosaccharide. That is more than 1000 monosaccharide. Okay, so let's see the structure of some polysaccharide. Okay, let's see polysaccharide. Okay, now when you're talking about glucose, okay, that is cellulose, for example, cellulose. Cellulose is made up of when glucose repeated several times. Okay, glucose is the monomer of cellulose. When glucose repeated several times, then it forms cellulose. Okay, now all the glucose are linked by glycosidic bond, and it forms cellulose. Okay, that's why cellulose is called homopolymer, because single type of monomer are repeated several times. Okay, in the same fashion, when talking about inulin, that is a polymer which is made up of fructose. Okay, when the fructose repeated several times, just like a cellulose, then it form inulin. Okay, that is glycosidic bond. This is linked by glycosidic bond. Okay, so the cellulose is the repetition of glucose and the inulin is the repetition of fructose. Okay, please do remember and mark this point. This is very important. Okay, now when the glucose is excess, for example, when glucose is excess in case of animal, okay, then it is called glycogen. Then it is stored in the form of glycogen. Okay, but when you're talking about a plant, if the glucose is excess in plant, then it is stored in the form of starch. Please do remember, it is stored in the form of starch. That is plant in the form of starch. So let's see the structure of glycogen first of all. And glycogen may be branch. Please do remember, it may be branch just like this. Okay, so it contains two end, that is left and right end. Okay, right end is called reducing. That end will be reducing in nature. Okay, and left end are non-reducing. Okay, now let's see the structure of the starch. A starch exists in a secondary structure. Okay, as I have stated that when the linear chain coil upon itself, please do remember, when the linear chain coiled upon itself, then it forms secondary structure in the form of helix. Okay, so the crevices, pocket and loops are created in which the starch can hold easily I2 molecules. Okay, it traps I2 molecules in it, due to which it appears blue in color. So it shows iodine, a starch iodine test. Okay, but when you're talking about cellulose, the cellulose is a linear chain of polymer. Okay, it is a linear chain of polymer. It do not have a fault, crevices and loop. That's why it do not trap the iodine molecules. That's why it do not gives any color. Do not gives any color. Okay, now when you're talking about cellulose, that is paper made and silk fiber. Paper made and silk fiber. Okay, then all these are made up of cellulose. Okay, in the same fashion we're talking about a bacterial cell wall, that is made up of peptidoglycan. Okay, it is also called murin. Then it is made up of two subunits, that is N-acetyl-muramic acid and N-acetyl-glucosamine. Okay, in the same fashion in the exoskeleton of arthropods, or when talking about a fungi, then the cell wall is made up of chitin. All these polysaccharides are homopolymer. Okay, mainly most of the polysaccharides are homopolymer. Okay, now thank you guys for watching my video. If you do really like this video, then please subscribe the channel and share with your friends.